Okay, I hope everybody can see this. So just a brief outline. I will talk briefly about what we know about microRNA and a couple of milestones in general in the microRNA field and as they relate to the immunology field. Then we will talk about a couple of recent overview articles, review articles, current methods that you can use to study microRNAs. There we will focus on microarrays as well as next-gen sequencing. And then the last part will be covering a couple of publications. I think there are 8 or 9 different publications that we briefly highlight. And as Chris mentioned, if you have any questions, just feel free and type them into the chat box as we go along, and they will be answering them. There will be replay. Also, you will get the PDF files and of the slides, so all the links and so on that I was referring to will be included in that PDF file as well. So let's get started about the current state of microRNAs, what we know. MicroRNAs, of course, small non-coding RNAs, usually around 20 to 22 nucleotides long, a little bit longer for plants, up to 24 nucleotides. They all derive from microRNA precursors that are very well defined. They have a stem loop as well as a hapless structure. And this precursor molecule is extremely stable. Many microRNAs are highly conserved, even between different species, for example, from worm to human in animals or in the plant kingdom. It's the same case. We know that they bind complementary to messenger RNA molecules and act as negative regulators of the translation. So and there are 2 different mechanisms depending on whether you're looking at plant or mammalian systems. I will mention that on the next slides. They exist in a very high copy number, and what makes them so interesting for biomarker development, etc., is that their expression is tissue and developmental stage specific. As of today, there are more than 25,000 mature microRNAs in the public database called MIRBase. There are different versions released every year. Initially, the pace was really incredible. There was an update pretty much every 3 months. In the meantime, it has slowed down. So the latest update was in August 2012, and these microRNAs are covering so far in the database 193 plant, animal, and virus species. The mechanism is rather complex. It's complex because each microRNA can control many genes, and the other way around, each gene can be targeted or regulated by multiple microRNAs. And it's the estimate that we found in a paper from, I think, 2 years ago. The estimation was that roughly 1/3 of all human genes is being regulated by microRNAs. There are currently 2 hypothesized mechanisms. One is the complete pairing, which is predominant in plants. In this case, the messenger RNA and the microRNA that is regulating the messenger RNA is a perfect match. So we have complete pairing, which then results in degradation of the messenger RNA. That's one way of downregulation. The other one, which is typically seen in the animal model, is that the pairing is imperfect, so there's some mismatches, but the binding of the microRNA is strong enough to repress translation of the messenger RNA. So the messenger RNA translation gets blocked, but the messenger RNA remains intact. Looking at the milestones, a little bit at the history, everything started in '93 when Lin4 was shown to encode 2 small RNA molecules, which was completely out of the ordinary at the time, because back then the dogma said that genes should encode proteins, not other small RNAs. Then in '98, for the first time, RNAi is observed, and Andrew Fryer and Craig Miller won the Nobel Prize for this discovery then in 2006. So this work was done in worms. Yeah, and as I mentioned, this whole research from '93 to '98 really challenged the central dogma of biology, which said basically that DNA gets transcribed into RNA, and then the RNA has no other function than being translated into the protein. In 2000, for the first time, microRNAs were identified in humans and Drosophila. That was in Slack's lab. Excuse me. The microRNA was LAT7. Starting from 2000, the early 2000s, the pace really accelerated. 2001, then Bartel, Tursch, and Ambrose discovered a large class of small regulatory RNAs, and that's when the name microRNA was actually born. When it comes to the pathway and the biogenesis of microRNA, 2002 was an important date. This was when part of basically the whole pathway was developed. The processing from prior microRNAs via the Drosha complex to pre-microRNAs via dicer molecule to then the mature microRNAs. And as mentioned, all these links here, these abstracts that you see here in blue, these will be active links in the PDF file, so you can read up using the original articles. Important, of course, is also the technology that goes that accelerates the research. So 2004, for the first time, microarrays were used to profile microRNA expression. 2005, then already next-gen sequencing was used for the first time using Arabidopsis plants. So it was done back then with Solexa, which is now Illumina. And 2005, the Sanger Institute back then put the publicly available microRNA database online, and it's nowadays available at mirbase.org. Then further developments, 2008, and that's certainly also still true to today. The latest are that circulating microRNAs are becoming more and more important. So they were first discovered in in body fluids in 2008 in serum and in plasma. And of course, that opens a whole other opportunity of very non-invasive diagnostics, etc. So this is really critical. And then I should mention here also nowadays this continues into other body fluids like urine, etc. Looking at the immunology field, 2004 is certainly a milestone. The paper of Lodish and Bartel and Chen in 2004 showed for the first time a microRNA involved in cell line differentiation. This was microRNA 181A, and it shows that it can modulate B and T cell differentiation. And same year also a paper from Kalin showed that there are certain signature microRNAs that correlate with specific types of leukemia. 2005 then, several papers showed that microRNAs have an important role in modulating the innate immune system, especially microRNA 32 is here of importance. Basically, it was shown that it can counteract. Basically, it can help fighting a virus infection. Well, then 2007, from 2007 on, more and more publications showed that microRNAs are playing an important role in modulating not only the innate but also the adaptive immune response. So again, microRNA 155, you will see that show up quite frequently. Another one is microRNA 181A, as I mentioned already before. That shows also up a lot. So, and these are just a couple of papers that were important in 2007 and started the development of microRNA research in the immunology field. This is a slide where I typically spend too much time on, so I will try to make it short this time. It's just really a rough outline about the microRNA processing. Basically, the gene is being translated into the primary microRNA transcript, or prime microRNA, which has a 5' cap, and a poly-A tail at the 3' end. This then gets processed via Drosha and Pasha, some enzyme complexes to the so-called precursor microRNA, which is extremely stable. All this is happening in the animal model, still in the nucleus, and this precursor microRNA then gets transported into the cytosol and further processed by a dicer molecule. So what the dicer molecule is doing is basically it's cleaving off the 3' and the 5' end here, as well as it's getting rid of the hairpin structure. So what you end up with is a microRNA antisense duplex, and this then gets cleaved into the mature microRNA molecules. And these mature microRNA molecules can then form RISC complexes, miRISC complexes, which are basically microRNA-induced silencing complexes. You see here the star mentioned. This is outdated, so nowadays this is the old literature. So, but I still see papers even this year being published with microRNA stars mentioned, or I should call it asterisks. It's basically the antisense microRNA, and initially it was thought that it's not important, that there's only one mature molecule, and now it turns out that actually both can play an important role, and the nomenclature was changed to use now -3P and -5P. And for any further information, especially the nomenclature, I can refer here to the microRNA to the mirbase blog. Sam, who's maintaining this blog, is doing an excellent job in explaining it. There's an article called "What's in the Name," and this explains the latest changes, which were implemented in 2011 and finalized in 2012. Looking at the what we know about microRNAs and the number of microRNAs known and well characterized in mirbase, this is the historic development. We left out here version 1, but it basically starts in 2005 here with version 6 and all the way to the latest version 19, which was released August of last year. As mentioned, there are more than 21,000 mirbase entries. These are the precursor molecules, and as mentioned, there are more than 25,000 mature molecules, covering more than 190 species. You see here nicely in 2008, that's when Illumina released its first small RNA sample prep kit. You see there's an even faster increase in known microRNAs. So next-gen sequencing was certainly crucial for the pace that this field has seen. Same is true for the microRNA publications. If you look at the related PubMed publications, it's almost an exponential increase over the years. And in 2007 or so, you would still meet people at conferences that would doubt the whole microRNA field and the fact that they exist, but lately I haven't found that at all. Latest from the funding agencies here in the US was just last week a paper or announcement that there's funding available for small businesses developing a biomarker platform specifically in the immunology field, in particular the progression of inflammatory diseases. So if you're in that field and you're in the US, this might be something to look at. So why would you study microRNAs in the immune system? There are, of course, a couple of reasons. First of all, microRNAs are, of course, tissue and developmental specific in their expression levels. And this is also the case for the immune system. For example, in the thymus and the bone marrow. Many microRNAs have been found to be important in the differentiation, development, and so on of B, T and T cell lymphocytes, dendritic cells, macrophages, and other immune cell types. And the publications that we will briefly highlight will show you that too. MicroRNAs are clearly participating in the modulation of both the innate and the adaptive immune response, and microRNAs are also involved in the inflammatory response. To dig down a little deeper, so dysregulation of microRNAs can contribute to the development of immune-related diseases. First of all, inflammatory diseases such as arthritis, atherosclerosis, any type of inflammatory disease. Then also autoimmune diseases, for example, as relatively harmless like eczema, and as severe going down the road to lupus. And also it plays an important role in leukemia, in cancer, blood cancer. Circulating microRNAs are becoming more important, I mentioned, so they of course will be great biomarkers for immune-related diseases as well. And microRNAs that are associated with these immune-related diseases are of course potential therapeutic targets, or even antiviral therapeutic targets. And this graph is basically just it's a very simple graph, but I liked it. It's show what abnormal microRNA regulation can affect the innate immune response, adaptive immune response, immune cell development, B and T cell function. This is a overview or review paper published in Autoimmune Reviews, which I just wanted to briefly highlight. So we just put it in here so that you can take a closer look at it later. But basically, it summarizes the current state. It's from 2009, but it's still current enough. Here in this table just summarizes the different microRNAs, their expression site, their targets, gene targets, and the function. And then, of course, the references. So I mentioned already miR-155 plays a big role, especially also in inflammation. miR-181A, 17 to 92, they both play a role in B and T cell development. 146 will show up later again. It's important in activating monocytes, and miR-223. And this is just a nice, from the same paper, a nice schematic overview where which microRNA is involved. Here at the beginning, at the monocyte development, both 146A and 155 are involved, and then here miR-223 plays an important role. And the interesting thing is that the same microRNAs that are playing a role here at the beginning also, namely 155, plays an important role here at the end in the lymphoid system again, at the T cell regulation. So this just as another overview. If you look at the innate immune response, regulation of toll-like receptors and toll-like receptor signaling and cytokine response is important, and miR-155 that I already mentioned, 146 and 223 are playing a major role, as I just showed you on the slide. They're microRNA modulated pathways that are really critical here, and other important factor is the response to virus infections. So cellular microRNAs not only alter the immune cell development and function, but they are also able to directly affect viral replication. And there's actually a case study in one of the papers with the swine-related virus that I will show. Another point is that virus-encoded microRNAs can shape the host-virus interaction and regulate the viral life cycle. So you have the microRNA regulation in the host as well as in the virus itself that you have to look at. This is another review paper here, specifically dealing with the innate and adaptive immune response, in which microRNAs are involved here. So I won't go through all of them again individually, but you get the idea. A whole variety of microRNAs, their targets, and the effect that they have. Now, looking at the adaptive immune response, they regulate the toll-like receptors as well as the signaling pathways. MIR181A, which was one of the first one to be shown to play a role in the immunology field, can function to modulate the strength and threshold of T-cell receptors, their signaling, and by doing so they can influence the T-cell sensitivity to antigens. They're crucial in the antigen presentation and T-cell receptor signaling, and just overall they play an important role in the differentiation, proliferation, and activation of both B and T cells. And here is again from the same paper, this time a table of microRNAs that are specifically involved in the adaptive immune response. Of course, of interest is really, besides doing the basic research, the clinical potential, how we can help patients. And if you look here at the diagnostic field, there are the biomarkers in body fluids that would be very helpful for serum plasma, from serum plasma or the exosomes. It would allow to make accurate and detailed clinical diagnosis, and beyond that, it would also, or in certain cases, already does allow to determine the prognosis and predict the treatment efficacy. And in this context, there's already a diagnostic kit FDA approved on the market, which is by Rosetta Genomics. It's called MirView. And it's an assay to differentiate between 4 different main types of lung cancer. And before it was difficult for the clinician to make a clear diagnostic call, and with this tool, with this kit now, it's the success rate and the diagnostic accuracy has dramatically improved. And if you go beyond the purely diagnostic part, of course, microRNAs are becoming increasingly important in the drug discovery and therapeutic arena. So you can think about microRNAs that as immune-regulating drugs on one hand, or microRNAs on the other hand as drug targets. So you would basically prescribe an anti-microRNA as drug. And just in general, to microRNAs are of course interesting to understand any type of response to infection, stress, or other stimuli. When starting out an experiment or you're planning out your research, you're of course looking at a couple of different methods that you can use. From qPCR, arrays, next-gen sequencing, etc. And one really helpful paper I found is this Nature Review paper here. It's simply called MicroRNA Profiling, Approaches and Considerations. And it's from last year, pretty much exactly 1 year. And it's a very good paper. It helps you to come up with the best method for your sample type also, and in general what type of question you're trying to answer. So basically it walks you through, depending on what type of specimen, sample specimen you have, what type of method is available to you, and simply what you need to pay attention to depending on which method you're using, because there's no better method than the other. It's just depending on what you want to look at. So each method has its advantages and disadvantages. So this was a really good overview paper if you're new to the field and trying to figure out which method you should use to answer your question. And if you're looking at research tools that are available right now, starting out with MicroRNA Identification, there are of course direct cloning and sequencing methods. Next-gen sequencing is certainly the biggest there to find new microRNA sequences, but there are also computational tools that allow you to predict microRNAs, like miRNA-seq, find microRNA, miRScan, miRalign, and so on. You can do tiling microarrays, and as mentioned already, next-gen sequencing. That's certainly the biggest here nowadays in this category. Then once you've discovered the microRNAs, and if you're working with well-characterized species, the first part probably doesn't really apply to you, but the second part now, when it comes to detection and the profiling of your microRNA content, of course an old-fashioned method is northern blotting. Then you could do in situ hybridization, real-time PCR. But if you want to look at really the entire microRNA if you want to call it that, certainly microarrays and next-gen sequencing are the way to go. And we will highlight these two further in a little bit. So then you have determined your differential profile. Now you want to look at, which microRNAs are up and down regulated. Now you want to determine what these microRNAs target. That's certainly still a field where there can be improvement made. There are some bioinformatic tools, but like TargetScan, Picta, PETA, and so on, and many others. But if you ever use them, you will see that they don't necessarily agree at all. So there's still a lot of room for improvement. Then, of course, if you look at experimental methods, you can look at the gene expression levels from the same sample. You can look at proteome expression analysis. You can do pull-down assays, 5' race analysis, and specifically for plants, something that's becoming more and more popular is deglidome sequencing. Then going on to the pathway analysis, there are a couple of tools available like miRFocus, miRPath, and so on, but they're either relying on predictions, or if they're curated, then oftentimes they're certainly not based on the latest versions, miR-based versions, that is. So there's certainly certain lag time. Then there are commercial solutions available in the meantime, like Ingenuity has a software package that is certainly seems to be up to date. Then the final stage, looking at the functional analysis, you can, of course, probably the most common thing is, I guess, to use a knockout model. You can use Lucifer arrays assays. You can use antagomeres, so anti-microRNAs, to inhibit microRNAs, or you can use artificial microRNAs, or microRNA mimicry, synthetic microRNAs. Before you do any experiments, of course, you have to prepare your sample. And there's, in the meantime, a whole variety of solutions out there to isolate good microRNA or total RNA from your sample. It really depends on what type of sample you're starting out with. It's basically shown here from the most simple starting point, having a cell line, or even better, a real tissue sample, all the way to, of course, body fluids, to FFPE samples. These are 3 kits that we recommend that we have good experience with. The first one is a phenol-free extraction method. It's a kit from Norgem Biotech. This is a general one. Then the miRNA kit from QIAGEN, not to be confused with a regular RNA kit. So if you go with QIAGEN, make sure that it has this MI in front of it. And the miRNA kit from Ambion. And all three companies have specialized kits for, let's say, FFPE samples, or blood and serum or plasma extraction kits. So basically, go ahead and pick the right kit for your sample type. And important is, no matter which method you're using, make sure that you stick to it. Don't switch the kit in the middle of the experiment and process all samples using the same kit, because they all work, but they will all give you a slightly different profile. And just to mention in terms of sample prep, of course you can also use Trizol, but we've seen that people that are new to the microRNA field, they typically struggle a long time to get good results with Trizol. That's why we started recommending the other commercially available kits. You can check your sample quality by looking at the UV-Vis ratios. So the 260 to 230 ratio should be above 1. The 260 to 280 ratio should be above 1.8. Of course, that's just really a pretty crude estimation, but you can also run a bioanalyzer gel. We don't really routinely do that because we've seen it over and over again that the RIN values that you're getting with the bioanalyzer are, of course, no quality measure for the much smaller microRNAs. It gives you a good overview picture over the sample quality, but we've seen it where you had a RIN value of 10 and the microRNA was completely degraded, or the other way around. A RIN value, the lowest we had was 3, I believe, and the microRNA was just fine. So unfortunately, there's really not the perfect QC method out there yet, at least not high throughput, because you kind of run it on a gel. These are just some samples, courtesy of Norgem Biotech. What are shown here are basically 2 different sample preps, a good one and a poor one. The poor one is either could be where the microRNA is completely degraded. You see it's completely missing here. Or in this case, they used actually a sample, total RNA extraction kit that was not suitable for the sample type. So assuming that you have now a decent total RNA sample, so you don't necessarily have to fractionate it into the large RNA and the microRNA. We, for example, work with total RNA samples that customers send us, and pretty much everybody else does the same thing. Now looking at the methods that you could use for the differential profiling, there are the microarrays, of course, and then the RNA sequencing, or next-gen sequencing, and they have both different advantages and disadvantages, of course. So let's look at the microarrays first. It's a proven method, has been around for a while. It's pretty high throughput. You can analyze really hundreds of samples per month Of course, you look at all the microRNAs, the known microRNAs at the same time. Yeah, it's pretty straightforward in terms of data analysis, and turnaround time if you're using a service can be pretty quick, as fast as 5 days. If you're doing it yourself, of course, even faster, and it's fairly low cost. Disadvantage, of course, is really that you can't really use it for discovery. You're looking at the known microRNA content. And so let's look at this a little bit closer. This is just, I'll pretty much skip over that. That's just the workflow here in-house. What happens, what we do when we receive customer samples, basically QC, sample prep, etc. But excuse me. Basically, you're running your different you experiment with let's say a control versus a treated sample group. So you run you have a couple of biological repeats. You run the assays, the microarrays. This is the overall heat map showing you all microRNAs and all samples. And then you apply some simple statistics to look at those that are up and down regulated. And here's basically the result of a t-test shown at different p-value cutoffs. And the p-value is, of course, the measure for the statistical significance. So the lower, the better. So these microRNAs here are clearly differentiating between the control and the treated group. So that's pretty straightforward and fairly low cost. If you're looking at sequencing, of course, it's really has the major advantage that it's not limited to known microRNAs. So of course, it's ideal for discovery. Besides microRNAs, you could also look in a different sequencing run for messenger RNA, long coding, non-coding RNA, or of course also shorter non-coding RNA. So it's a truly quantitative method where you get relative expression levels, but with array sequencing will give you actual copy numbers. So it's truly quantitative. It has an increased dynamic range, and you can also detect structural variations such as gene fusions or alternative splicing events. This is just, again, the workflow. I will skip over that. So to give you some ideas about the numbers, currently for the small RNA sequencing or microRNA sequencing, we're using the Illumina Genome Analyzer GA2X, and the read lengths are 35 bases. Typical, depending on the sample quality, you get 20 to 30 million reads per lane. And then it's pretty much up to you how many samples you will put on one lane. So, of course, the number of reads per lane stays constant. So if you use one sample on this lane, you will get really very deep sequencing data, 30 million reads for this sample, which will certainly allow you to even detect very, very low abundant microRNA. So for discovery purposes, we would certainly recommend that. If, on the other hand, you are just looking at differential profiling, you could go with, let's say, our recommendation is to use not more than 3 to maximum 4 samples per lane. So this would then still give you up to 10 million, let's say 7 to 8 million reads per sample. If you go below that, we don't really recommend that. Then you're losing out on low abundant microRNAs. So for discovery, certainly the best way to go. For differential profiling, we feel at this point that if you're working with a well-characterized species such as human, mouse, rat, etc., it's certainly not only more economical, but yeah, it's more economical to go with microarrays at this point. But it's also it's less biased in terms of sample prep, because if you're working with microarrays, you're pretty much using your total RNA sample pretty much directly. versus sequencing, where you first have an extensive library prep with amplification steps, etc., where there's the chance that you're biasing your profile. Another important thing when it comes to sequencing is we've noticed that a lot of people interested in microRNA sequencing, they think that sequencing is such a cool method that you don't need biological replicates. But of course that's not true. It doesn't matter which method you're looking at, you need to have the same number of biological repeats, meaning minimum really of 3 to be able to do any statistics. If you're just comparing one control versus one treated, and you see a difference, you have no idea whether this is really due to the treatment or just random variation. This becomes, of course, more important as you go to actual patient samples. So where 3 biological repeats are good enough for to get some initial data for, let's say, cell line or animal tissue data. When you're dealing with patient samples, the patient-to-patient variation between the control patients is already so big that depending on how narrow you can focus on a patient group in terms of gender and age and disease state, you're looking at the very, very minimum at 10 samples per group. And we had recently a case where reviewers rejected a paper where the author had 30 samples per group. They wanted 50. Yeah, besides that, it's important to have ideally the same number of samples per group. This is just a brief overview over the data flow, basically the data analysis part of the sequencing data that we're generating. So by default, we're delivering raw reads, of course, as well as mappable reads. And then our proprietary software goes through and basically classifies the sequences that are generated into a couple of different groups, 4 different groups, or 5. Group 1 are the known microRNAs for the species. Then group 2, known candidate microRNAs for this particular species, and then all the way to, of course, no hits whatsoever, and group 4, of course, the most important one, potentially novel microRNAs. And this is just a tiny sample of a table that you get when you have sequencing data. And it's just to highlight that there are isoforms for each microRNA. So you see here the total number of reads for this particular microRNA molecule. It's almost 60,000 reads. So clearly the most abundant one. But interestingly, you see here a second one that is shortened by just one nucleotide, which still has a pretty high read count of almost 11,000. And then you have other ones that are less prominent, really just a few copy numbers, but they're either shifted or truncated from one way or from one end or the other. And so this phenomenon is called these are called isomers. And knowing this, it explains can explain some of the discrepancies that you observe when you look at different methods. For example, if you have array data, the array probes will be, of course, made against the sequence that is in the database, which will be this one here. And but the second one here with 11,000 counts will still bind to the probe because it's just truncated by once. Versus qPCR, for example, which will differentiate between these two. So that's an easy explanation between for some of the discrepancies that you see between methods. Moving on to some of the publications. Basically, I will highlight really just briefly papers from 3 different categories. One is the general differentiation or general immune-related. Then I'll move on to innate immune system related, and then going on to the adaptive response, how microRNAs are involved. So the first paper here is from last year, is talking about how microRNAs might be really crucial in the immune in the development of the immune system for newborns. It was shown that immune-related microRNAs are abundant in breast milk exosomes. There are a total of this was done all with next-gen sequencing. So there are a total of 452 precursor microRNAs found in breast milk exosomes, which then result in 602 unique mature microRNAs. And out of these, 59 are present and enriched in the breast milk exosome. It's very interesting to note also that there were experiments done about the stability of these microRNAs, and it was shown that these 59 immune-related microRNAs are very stable and as compared to synthetic microRNAs. And so the authors of this paper postulate or propose that some of these, if not all of these, immune-related exosomal microRNAs are allowing basically the mother to transfer genetic material to the infant and enhance the immune system and aid the development of the immune system of the infant. Then when it comes to differentiation and development of and how microRNA relates to it, this is a case where from this year publication where the authors looked at the differentiation of human peripheral blood monocytes into macrophages, and they've shown that it can be reproduced ex vivo by just culturing the cells in the presence of CF1, which stands for colony stimulating factor 1. In this case, microRNA arrays were used, and basically you see that here in table B that once you add CF1, the expression of microRNA 142-3P in human primary CD14 plus monocytes dramatically drops over the period of a week. And they've done also the experiment where they reinstated expression of microRNA 142-3P, and they were able to show that this will then reinstate basically the differentiation of these cells. So the downregulated version here that you see here is so this abnormal low expression of 142 is something that you see in monocytes from patients with the most proliferative form of chronic leukemia, or CMML. So that's the background of this experience, of this experiment. This is a paper from Lukev's group, also just published in 2013, a neuro report, and it shows the expression of microRNA 34A and which role it plays, because this is a microRNA that's of particular interest for Alzheimer's patients where it is upregulated. And so looking at microRNA 34A, they found that it's actually targeting the 3' UTR region of a gene called TREM2, which stands for triggering receptor expressed in myeloid cells 2. And basically, this is shown here, the sequence of microRNA 34A, and then the 3' UTR region of the TREM2 gene, and how it matches. As mentioned in the mammalian system, it doesn't have to be a perfect match, but this binding is clearly strong enough to inhibit expression. Then here is just shown a heat map that is the result of, again, microarray data here, looking at the most prominently differentiated microRNAs. So 34A, 125B, 146A is a very frequently discovered one, 155 as well. And then pretty much as a control, 183, which doesn't change at all. And they've done this with microarrays as well as with northern blot. And it's a great agreement. And yeah, so these results are hint that that 34A, microRNA 34A is clearly involved in downregulation of TREM2, and yeah, it's important in the inflammatory neurodegradation. Looking at the innate immune response and how microRNAs fit in there, this is a paper also just published, Journal of Virology, 2013. They did a time course study over 3 weeks with pigs, and looking at, so basically they infected pigs with a virus that is one of the biggest problems in the swine industry. It's an important viral pathogen. It's called porcine reproductive and respiratory syndrome virus, or PRSV. And they looked at what happens if you infect the swine and the pigs with the virus. So that's the control group here in black. And you see the temperature rises, so there's inflammation going on. And yeah, after 2 weeks roughly, most of the pigs were dead. And then they tried to a treated group where they basically treated them with microRNA 181. And as you can see, it's the temperature rise is much delayed, and the pigs are actually living longer by roughly a week. And the other graphs here just show you the viral load in the pig animals. Again, black the control, and then how it looks with the microRNA in induced pig. So this is clearly showing that microRNA 81 is playing an important role in the host's microRNA response to PRSV, and that this might be a good target for antiviral therapeutic development. Moving on to another economically important species is in Asia are the Chinese midden crabs. They are the most important freshwater aquaculture crustaceans in China. And again, it was basically looking at the microRNA response of a control group versus the infected group. And this Venn diagram here nicely shows the microRNAs that are expressed in the normal sample group versus the infected sample group. And you see that 95 are downregulated in the normal versus infected, and 133 are upregulated. And out of these 133 upregulated microRNAs, 87 are really unique to the infected species. 46 are expressed in both the normal versus the infected. And again, this might be a first step basically into developing some kind of drugs or to find some drug targets for this particular disease. I will briefly skip over that one because we're running out of time also. Looking at the innate immune response regulation, here this is a paper also just published looking at LED7G and how it regulates expression of inflammatory cytokines. The experiment was done with sheep muscle, fetal sheep muscles, and the microRNA expression levels were analyzed using microarrays. Beyond that, they looked at the tumor necrosis factor alpha and IL-6 messenger RNA expression, both of which were higher in an obese group compared with the control tissue, and which indicated an inflammatory response in obese fetal muscle. Yeah, overexpressed LED7 in these cells result in a decrease in the expression of inflammatory cytokines, TNF-alpha, IL-6, as well as TLR4. And so this is why the authors conclude that microRNA LED7G has probably a role in the development of chronic inflammation in the fetal muscle of obese sheep. And here just the diagrams just show the up and down of control versus obese group. And then moving on to the adaptive immune response regulation, this is looking at the immune regulation inflammation processes in the early phase of, and I have to excuse myself, I really don't know how to pronounce this right, schistosoma, I guess, japonicum, or better known as snail fever or bilharziosis, which is still a really important public health problem that affects 200 million people in 76 countries. And basically what was looked at here was the microRNA expression difference between in mice, in tissue of mice, before and 10 days post-infection. And they looked at the different tissues, the spleen, lung, and liver. And yeah, again, looking at the Venn diagram here, that resulted in the result of the analysis. You see that 132 microRNAs are consistently differentially expressed, no matter which tissue you're looking at. So that's clearly a great step in developing a clear biomarker signature profile. And the authors went on and did some pathway analysis, and yeah, the results are clearly helpful in developing a potential new drug for this disease. Looking at the inflammation process and how microRNAs are involved here, very early on, I mentioned already microRNA 155. This is one of the many examples that it's involved here. This is specifically looking at the myocarditis and how microRNAs contribute to the inflammatory response to viral infection of the heart. This was done again with microarrays. And here you see the time course experiment in the heat map. This is the control group, day 4 and day 7. Here's the tissue samples, of course. And you see how this group here clearly gets overexpressed. And so what was found is that microRNAs are clearly upregulated during inflammation. And if you add an LNA-based anti-microRNA 155 molecule, it clearly helped the inflammation. Basically, it toned down the inflammation. So the bottom line of this paper is that 155 is a potential drug target against to help with inflammation. So this just as a brief overview. Just in case you haven't heard about us, we are based in Houston. We are the first microRNA array service provider, so we've been doing this since 2005. Processed a lot of samples. Over the years, we started out with offering our own proprietary microarray platform, which is very customizable and flexible. That's why the microRNA field was a perfect research ground for us, because it's such a fast developing field, so we can keep up to date with the current developments easily. Yeah, we have a lot of customers in academic institutions as well as in the industry. We have customers from more than 40 countries. Tons of customer publications. You can find them on our website. There are more than 600 by far. And we have representations in also a big office in China, as well as representations in Japan, Korea, South Korea, of course, and India. European customers are usually sending us the samples directly. There's really no shipping issue. And so, yeah, that makes everything much easier. Other than that, yeah, I mentioned we started out with microarrays. In the meantime, we offer, of course, next-gen sequencing services as well. So this is just a representation where our offices are. Our representations, all samples end up here in Houston. All experiments are being done here in Houston. So the sequencing or the arrays, everything is being done here at the Houston location in the US. So with that, I thank you.